Thank you. So once again, thank you very much for the invitation. I'm really sorry I couldn't be there in real life, so to say. A little bit hard still for us to travel. Okay, sorry. This is going to jump. I see already now between slides for some reason. What I'm going to do today is that I'm going to tell you a story or two different stories about resident memory T cells and CD8 T cells in HIV infection and SARS-CoV-2 infection. So basically, this is going to be divided into two different parts. Number one on HIV and number two on SARS-CoV-2. So I'm going to start with part one now, which is going to be on much of the work that I did when I was a postdoc at University of Pennsylvania, but also some of the work that we've been continuing doing in my lab since then. So I think it's, during the course of a lifetime, we are exposed to a multitude of viral infections. Some are going to be more benign in nature, some are going to be more dangerous for us in general. And our immune system needs to keep all these, of course, at check. And one of the infections that most of us here are interested about is HIV. And we know from old studies, but particularly, I guess, from this study that came out a couple of years ago from Jake Estes' lab, where they assessed the number of HIV-infected cells or SIV-infected cells in rhesus macaques, and basically could calculate that over 98% of SIV RNA-positive cells are present within lymphoid tissues or lymphoid aggregates, particularly in lymph nodes and gut, but only 0.2% of the viral RNA-positive cells are present within the blood. And the cells that I've been interested about during most of my career are CD8 T cells. They're also known as the cytotoxic T cells. And these are really critical to maintain viral control, for example, of HIV, but also to eradicate many infections, such as acute viral infections. But in general, the problem is this: because although most lymphocytes are present within tissues, the vast majority of the knowledge of human CD8 T cell biology is derived from studies of the blood. And this picture basically, I think, summarizes pretty much what my lab is interested about, is really trying to understand what the location, function, regulation differences are between virus-specific cells that are present within tissues versus blood. And why is this particularly of interest? So it spells to a high degree resident memory T cells, because these cells we now believe are the sentinels of our immune response. And these cells do not recirculate at steady state, and are usually described, at least in humans, by expressing CD69, and they can also express other markers such as CD103, CD49A, etc. And particularly when it comes to CD69 expression, for many of us, it has been known to be an early activation marker. And to some degree, of course, it is, because when you activate T cells, it comes up. But really, its functional role is to downregulate S1PR1, which is the egress receptor that thereby prevents T cells to leave tissues. So that's really its functional role. And we know from really beautiful studies from Dave Masterpost and others that Trams really outnumbers the circulating CD8 T cells in non-lymphoid tissues. This has particularly been shown through parabiosis experiments in mouse, where you can look into different types of tissues, such as small intestine, pancreas, etc. You can really see that most of the CD8s there, memory CD8s, are of a resident are of resident characteristics. However, if you start to look into secondary lymphoid organs, you usually do not find resident memory T cells to the same degree at these sites. But one thing that is important to remember, as you can see from the citation here, is that back in 2015, when people still, including Dave Masterpost's lab, were using specific pathogen-free mice to study residency. So basically, instead, if you look into Trams in lymph nodes and you start to compare steriles, in other words, specific pathogen-free mouse versus co-host mice, and even pet store mice, you start to see this pattern that you get more and more memory cells based on CD44 expression, but you also do see that increased levels of the Tram marker CD69. And what was very nice of Masterpost's lab, and what they showed at the same time point, is that if you basically tattoo in antigens like OVA and you stain for Synfecal or GP33 by tetramers, you do actually identify that within in the draining lymph nodes in mouse, you do see TRMs coming up with this specific canonical expression of low CD62L and high levels of CD69. But in general, you do not see them in non-draining lymph nodes, at least to a high degree. So really what this tells us is that expansion of TRMs or CD69 positive CD8s in lymph nodes take place with immunological experience and close to the site where antigen exposure takes place. So this, to some degree, set the status for some of our work that we did on HIV back in 2018, when we started to characterize lymph nodes from HIV negative donors versus HIV positive individuals. And really what we could find here, one of the early striking features was the fact that the frequency of CD69 positive cells, and particularly those cells with low levels of other egress receptors such as CCR7, they are enriched or higher, at least, in terms of frequencies in HIV-infected lymph nodes compared to HIV-negative controls. Further on, what we did were to characterize cells, in particular HIV-specific cells, between blood here on the top and versus lymph nodes here in the bottom. So essentially what we did were to stain HIV-infected blood versus lymph nodes with tetramers here. This is data flow plots from the same donors, match blood versus lymph nodes. And in general, what you can quite easily here experience is the fact that HIV-specific cells in lymph nodes have to a high degree CD69 expression, and they have a classical TRM phenotype. We have shown that at least by transcriptional data and other type of data as well. And it seems to be that these cells are dominating the cellular response in lymph nodes. Another striking feature that we found at the same time point is that this is not only true for individuals which are non-controllers or on ART, it's actually really true for HIV elite controllers, and actually to a much higher degree even. So if you compare here, same thing here, gated on CD8 plus T cells, we do comparisons of CD69 expression on the y-axis versus a tetramer here on the x-axis. You do see that you have higher levels of CD69 in lymph nodes versus blood, but you actually do see that these individuals have these really high magnitudes of virus-specific cells against one epitope sometimes even that have a specific HIV-specific TRM phenotype. And as a matter of fact, what you do find here as well is that the HIV-specific lymph node to blood ratio is higher in HIV elite controllers versus those not controlling infection. And finally, the percentage of lymph node CD69 positive HIV tetramer positive cells are higher in elite controllers versus HIV positive individuals that are on ART or not. So that kind of lead us into a second story, like a follow-up story that we did at the same time point, where we wanted to further understand some of the functional mechanisms that could be behind elite control in these individuals. So essentially, one of the first things that we did here were to do simple confocal imaging stainings. We looked for HIV RNA in here, just simple pictures in elite controllers versus chronic progressor. And while you see very few dots here which are present with HIV RNA, you really don't see it, and you don't see it really in the vesical follicles. You do see it, on the other hand, in chronic progressors here, term CP. So really the levels of HIV RNA per million cells seems to be quite similar in lymph nodes in elite controllers versus ART, but much higher, of course, in people not controlling the infection. So furthermore, what we did here were to do more functional experiments, more mechanistic experiments, where we basically infected CD4 positive T cells from lymph nodes with HIV BAL, and then we co-cultured them together with different ratios of CD8 positive T cells. And when you look at just HIV together with HIV-infected CD4s, you do see that the quite high frequency of the cells are infected. But when you add on CD8 plus T cells, you see that they start to suppress the virus. So this has been shown before in the blood, but we could actually recapitulate some of these findings in lymph nodes. And you can see that the P24 reduction is better and higher for individuals controlling the infection versus those individuals not controlling the infection. And this is also true versus AR3 treated individuals. So in other words, lymph node CD8 plus T cells from elite controls can efficiently suppress HIV, but what does it correlate to? And one of the first things that we started to look for, and which was one of the hypotheses of this study, was that potentially these individuals have higher cytotoxic capacity. measured, for example, here by peripheral and Granson B expression, but actually turned out to be a little bit different than that. And instead, what we started to see was a positive correlation between the levels of peripheral and Granson B expression on memory CD8 positive T cells in general and increased viral loads. Because what we could do here was to also include acute HIV infected donors and lymph nodes from these individuals. And these individuals had really high levels of Granson B and perforin within those lymph nodes, which was then associated, if you combine all the data points together, with the increased viral loads. So really, what it seems like was the cytotoxic CD8 positive T cells defined by perforin and Granson B expression are almost absent from lymph nodes of elite controllers. But actually, this was something at this time point that we started to see was something similar between when you start to look into other tissues as well as steady state, also in healthy donors. It seems to be a general lack of cytotoxic Granson B positive, perforin positive, CD8 positive T cells in many tissues, and not only in lymph nodes. So one of the questions that we wanted to ask at this time point were, could it be so that cytotoxic T cells, so peripheral and Granson B co-expressing cells, are recirculating and surveilling tissues at all during steady state? So that kind of led us to a study here where we started to dissect and cannulate out lymph from the thoracic duct in people. So basically what the thoracic duct is that it's the major lymphatic vessels that cells use to travel back to the blood from lymphoid and non-lymphoid tissues. And essentially when cells are moving, so immune cells, when they're moving out to blood and into tissues, they need to circulate back to the blood through lymphatic vessels and via this big lymphatic vessel known as the thoracic duct. And some people, when they go through surgeries, different traumas, it needs to be a leakage repair of the thoracic duct, and that's the reason why we could gain access to these samples. And then we initiated protocols so we could also gain thoracic duct lymph from HIV infected patients. This is actually how it looks like. You can collect it in vacuum tubes, and essentially you can phycol it just normally as you do with blood. And this is how lymph looks like. It almost has this urine-like feature to it. And if someone has, for example, been eating a lot of fat, it will look more milky, which is called chyle. And this is just a comparison to the same individual how venous blood looks like. So essentially what we did here were to understand, do we find cytotoxic T cells when you look into the thoracic duct? So these cells are circulating from our tissues back into the blood versus cells that are present within the blood. And essentially the answer is no. So there's very few cytotoxic T cells that recirculate from tissues and back to the blood at steady state. So this is just gated on memory CD8 positive T cells, perforin on the y-axis, Granson B on the x-axis. This is blood. This is the same individual on TDL. And as you can clearly see here, you have fewer of perforin and Granson B positive cells. And this is also very independent on which type of phenotype these cells have, because even TEMRAs and the few TEMRA cells that you find here has lower levels of Granson B and perforin expression in general within cells coming out from tissues in comparison to those that are present in blood. So the question with that is that, does that mean that cells then are recirculating at all? Maybe they're changing phenotype after all. It's really hard to do, of course, mechanistic studies in humans, but I think that through better sampling of patients, particularly patients also which are set on different type of drugs, it is possible to really do cool and mechanistic studies in humans as well. So what we initiated was a collaboration with other physicians which are treating MS patients with this drug called FTY-720. And this is a drug that inhibits tissue egress of T cells, B cells to some degree as well, by working as a superagonist for this S1PR1 receptor that I talked about before that CD69 can downregulate. So essentially what we did here were to take blood samples before FTY-720 treatment, 1 month afterwards and 6 months afterwards, to really see what happens with human CD8 positive T cells when you block cells from going out from tissues into the blood. And when you do this, what happens really is that your naive T cells and central memory T cells, of course, they drop in both when it comes to absolute counts, but also, of course, in frequencies. But still what you can see here is that you still have this population of effector-like cells, or TEMRA cells as we usually call them, present here within the blood. And this to some degree can be displayed here because the absolute counts of memory cells are going down, but then it kind of stabilizes even out to 6 months after people have put on this drug. And the reason because of that is because these cells that we just talked about, the cytotoxic cells, seems to, they increase in frequencies, as you can see here, but their absolute counts are retained within the blood for a long time, 6 months at least after FTY720 treatment. So basically what this means is that blood cytotoxic T cells based on perforin and B expression are retained in the vasculature. So finally, what we did in this story were also to conduct single-cell clonotype and transcriptome analysis. So essentially what we did here, this is just an example of C and B, but I will come to HIV soon, is that we sorted out C and B specific cells against one specific epitope, and then we focused in the analysis on one clonotype. And really, even at the clonotypic level, when you start to compare blood versus thoracic duct lymph cells, you see that the major genes that are coming out to be differentially different for the same clones even are these cytotoxic genes. And this is for C and B specific cells, but it's actually true as well when you start to look into HIV specific cells of different with different type of clonotypes. So really what this means is that that in our human body, we have parallel differentiation programs across tissues that is independent of the expressed T cell receptor. And we have clones that develop into these cytotoxic cells that are retained in circulation of some unknown reason. And we have also other cells which are recirculating through our blood or tissues, sorry. So the summary of this first part is that a majority of HIV-specific CD8s in tissues display TRM-like phenotypes and characteristics. HIV-specific lymph node CD8 from elite controllers can efficiently suppress HIV, but whether the mechanisms through classical killing mechanisms remain unclear. And finally, we show the cytotoxic molecule expression is primarily confined to distinct vascular CD8 positive cell population that's retained in blood following FTY720 treatment. And it could mean that these cells expressing, for example, high levels of perforin enzymes, so the classical cytotoxic T cells in human blood, might be a unique population with vascular resident properties. Okay, so that was part one. So let's move over to part two now, SARS-CoV-2. So this is going to be more new work. Some of it is already published. Some of it is just got published, I'm going to say. Okay, so just a very brief intro to SARS-CoV-2 and to the field in general, and when it comes to T cells. So T cell response against our coronavirus. So we know that memory T cell responses can persist for up to 17 years after SARS infection. This was shown by Bertoletti's group through it has almost become like a seminal paper already at this point, but it was published in a 2020 paper in Nature by his group, where he basically could provide could provide data on the people that were infected with SARS-CoV-1 still had detectable very nice T cell responses 17 years after their infection. And we know from other models that has been developed in the SARS field that airway T cells, known as resident memory T cells, most likely protects against lethal challenge with SARS in mice. SARS-CoV-2 specific T cells can also be identified in most patients with mild COVID-19. And also we know, I was jumping between here, sorry. And we also know that so-called cross-reactive SARS-CoV-2 specific T cells can be identified in 20 to 50% of unexposed people. I will come back to this point about cross-reactivity in pre-existing T cells later in this talk. Or actually already now, I'm going to say, and then later on again. So this was a study that we conducted, one of the early studies, actually quite very easy early spot analysis that we did just to characterize the SARS-CoV-2 specific T cell response. And really already at that time point, what we noticed were that when we started to thaw out samples from individuals that were unexposed, not infected, we could actually see that some individuals, not all of them, but some individuals actually had early spot responses against spike membrane. Now, sometimes nucleocapsid, but most of the times not. And these responses in general were much lower in comparison to what you find from a convalescent donor, which has SARS-CoV-2, but nevertheless, you could detect them in non-exposed and convalescent donors. And then there has been multiple other groups, which of course have been showing this, and SETI's group were showing this even before us. But one of the points that we provide in this paper was just a simple sketch on it, essentially, that if you have had more severe COVID-19, it seems like you have a stronger response in comparison to if you have mild COVID-19. Some people even that are exposed family members, we found surprisingly had T-cell responses. But actually, after that, there has been papers on that exposure to SARS-CoV-2 seems to generate T-cell memory, maybe even in the absence of detectable viral infection or even seroconversion. But then we could also see that when you start to compare the blue dots here, which are basically blood donors from 2020, they have more responses than what the 2019 donors have. But even the ones, like I said before, the unexposed donors do have about 1/3 of them have detectable ex vivo SARS-CoV-2 specific T-cell responses. So with that said, and what we found out at this point was also that it seems like cross-reactive CD4+ T cells that recognize SARS-CoV-2 are detected more commonly in peripheral blood of unexposed individuals than SARS-CoV-2 reactive CD8+ T cells. However, a large number of memory CD8+ T cells reside in tissues where they might harbor specificities that could provide localized immune responses to SARS-CoV-2. And why would that be of interest? Well, of course, for people which are interested and have been working on TRMs, we know that they are to some degree are sentinel defenders. And this was really, picked up even by this review for about a year ago by Crossreactive, Lipsitch, and others, which basically modeled how pre-existing immunity would have some type of impact on the course of SARS-CoV-2. And essentially, if you start to look into this model 3, where you have TRM cells in the upper respiratory tract, basically what they came to the conclusion of is that if cross-reactive or pre-existing T cells will play a major role to mitigate the cause of SARS-CoV-2 infection, these cells will most likely have to be present at various sites of infection. So again, the question is, do we have pre-existing TRMs, resident memory T cells, in the upper respiratory tract? So one of the ways for us to answer that question were to start to look into our liquid nitrogen tanks and start to look into people that we had collected samples from before the pandemic. And we were able to collect about 40 tonsils and paired blood from about 40 children and 41 adults, a little bit different age spans and genders. And you can see here that the year of the surgeries were between 2015 to 16 and 16 to 2018, so well before the COVID-19 pandemic were breaking out. One of the things that is really tricky when it comes to looking into tonsils or tissues in general is that if we're going to detect these type of responses, it's really hard to detect them by just using classical AIM markers like CD69 combined with 4-1BB, CD40 ligand, etc. Even with tetramers, sometimes it can actually generate background, and with tetramers, of course, we're missing a lot of responses. This is maybe not the best approach to do it, but the approach that we choose to do it to be able to detect sensitive detection of virus-specific CD8+ T cells and tonsils, and that was essentially by using classical intracellular stainings after 10 hours with viral peptide pools, which is gated on memory CD8+ T cells. And this is actually a donor, a non-exposed donor, a person with a pretty good SARS-CoV-2 cross-reactive responses tonsils that are responding against spike, nucleic capsid, membrane. But you can see here has beautiful EBV responses, particularly, I'll come back to the reason for why that's the case most likely, but also CMV and also to some degree OC43, which is one of these endemic coronaviruses that most of us have encountered at some point. So basically, this graph here summarizes the frequencies of 4-1BB and interferon gamma positive cells. And essentially what you can see here is that there's no question, for example, EBV, CMV specific responses in general are higher in non-exposed donors, but you do actually still see SARS-CoV-2 specific cross-reactive responses. And the prevalence of that differs quite a lot between different individuals. But if you do combine them, individuals that have one antigen or more, we do find detection of cross-reactive CD8 T cells that are present against SARS-CoV-2 in around 30% of tonsil samples. So what's the difference between if you look into SARS-CoV-2 specific responses in blood versus tonsils? So this is flow plots on the left side, which is a comparison between blood versus tonsils. This is DMSO, so this is negative background. And indeed, you do find when you look into the blood versus tonsil, higher responses in general in tonsils for the cross-reactive SARS-CoV-2 specific response. And this response in general, it's higher. This is just a chart showing that it's higher for spike, nucleocapsid. It doesn't reach out to significance for membrane of. EBV responses, again, much higher, in tonsils versus blood, while CMV seems to be the other way around, which I'll soon come back to why that's the case. And really what we found here was this fact that if you look at donors with detectable SARS-CoV-2 specific responses, you do find many more of these responses in tonsils versus blood, while the CD4 counterparts are quite similar between the two different sites. Next thing we wanted to do was to do some classical immunophenotyping by multicolor flow cytometry to really try to understand if some of the features that we just found was correlated to the fact that these cells have any kind of resident characteristics. And indeed, if you do UMAP plots like this and you overlay, for example, CD13 expression, CD69 expression, but also other markers like CXF5, PD1, etc., you do see that many of the SARS-CoV-2 specific cells, cross-reactive ones, but actually many other EBV ones as well, they do cluster here, down here, where CXF5 expression is pretty high, together with CD13 to some degrees and CD69 expression. On the other hand, as mentioned before, it seems like the CMV specific responses are higher in blood in comparison to what you find in tonsils. And they to a lower degree have are expressing tissue resident markers such as CD69 or CD13, and the ones which are expressing, as you can see here, highest levels actually of tissue resident markers are the OC43 specific cells. So about 50% of the SARS-CoV-2 cross-reactive cells are expressing CD69 and CD13, which is like these two canonical markers basically for TRMs. Finally, just to look at the functional profile of very specific CD8 T cells, more than just looking to interferon gamma, we can of course look into TNF, CD17A, IL-2, and many other functions. And right now, what we're doing in our lab is to extend this to do single cell TCR single cell RNA sequencing protocols and also secretome analysis. But one point I just want to point out there from quite simple analysis is that you can almost already hear the shifter that the frequencies, but also the magnitudes or MFIs of the interferon gamma response is a little bit lower for spike-specific responses in comparison, for example, here to EBV-specific ones. And indeed, if you start looking into the polyfunctional features here as well, you do find that, yeah, again, that the mean levels are lower for SARS-CoV-2 in comparison to most of the other ones, and that the polyfunctionality is lower for SARS-CoV-2 cross-reactive ones versus EBV, CMV, and OC43. So I think, again, what this means is that they are there, they're present, but they are less functional in comparison to what normal, so to say, primed specific cells are going to be in tonsillar tissues. So the conclusion of this is that SARS-CoV-2 cross-reactive CD8s preferential localized in tonsils if you compare it to the blood. SARS-CoV-2 cross-reactive CD8 T cells also show a tissue residency and follicular homing phenotype. And cross-reactive with cells with lower functional capacity, they have lower functional capacity than other virus-specific T cells induced through natural infection. So the question is still, which we don't know about, is whether the effect of pre-existing tissue resident SARS-CoV-2 specific cells are important for mitigating the cause of SARS-CoV-2 infection and for immunity. This is ongoing work in the lab. So actually with that, I'm done and really would like to thank people, particularly in my group, Takuya Sakina, which was instrumental for the initial SARS-CoV-2 work, And together with Andre Pires-Potti and Olga Rivera-Ballesteros, Julia Nasal is the one that generated the data here towards the end on resident memory T cells, cross-reactive ones, and also, of course, other people in my lab. And yeah, big thanks, of course, to my previous lab in the US at Penn, Mike Bett's lab, which basically spearheaded it, came up with a lot of the ideas for the initial work on HIV that I presented, that together with the great collaboration with John Berry's lab. Also want to thank surgeons at the Hospital of the University of Pennsylvania, David Price from University of Cardiff for tetramer conjugations, samples from Case Western, NIH with Danny Dewick, Samuel Dorica for TCR analysis, and also, of course, patients, healthy volunteers, and my funders. So thank you very much, and I'm happy to take any questions.
Thank you so much for a great talk. We have lots of time for questions. You can either put it in the chat or you can raise your hand and then I can unmute. So it sounds like David Cole has a question. David, I can unmute you.
I think I'm unmuted.
Oh, you're unmuted. Okay, cool.
Yes.
Hi, I don't know if you can't see me, but pleasure, wonderful talk and thanks. I sort of had a question about towards the end, you were showing the UMAP plots of the functional characteristics of the coronavirus specific T cells that you recovered by single cell work, I think out of the tonsils, and comparing and contrasting EBV, CMV, OC43, and SARS-CoV-2. And I was struck that the placement in the UMAP of the OC43 reactive cells, which presumably was from prior infection, childhood infection, and so forth, was different from the SARS-CoV-2 reactive cells, really quite contrasting. But yet there is an underlying hypothesis, I think, that cross-reactive cells are maybe placed there by prior infection. So how do you interpret the strikingly different location of the cells in the UMAP space?
Yeah, no, it's a great question. It's actually one of those questions that one of the reviewers were having to us as well. I don't think it was you. Well, I don't know, of course, but it's a good question. And I have to be honest to say that we don't have an extremely good answer to it, because we don't think that these cells are coming from EBV specific ones. So we do occasionally see, though, because some of these analyses, they're based on that we are collecting together basically data points from a couple of different donors, but not every single one that we ran in these analyses. But you are right about the fact that even if we see that both OC43 and SARS-CoV-2, they have resident phenotypes, it seems like they have a little bit different ones. And I don't really understand, we don't really understand why that's the case in general. So I agree about that, and I don't have like a full answer to it at this point, more than I don't think it's EBV's cross-reactivity that is causing it, actually.
So interesting, and I've got some technical stuff I can follow up with you later, but I just noticed if you look at that map, and it looks sort of like Asia, and there's Taiwan or something over on the lower right, the little island, where there's distinct cluster of both purple and red cells. Is that a particular phenotype that you can recognize from correlating with other markers of a certain, outstanding phenotype of T cells that's sort of down there in that little cluster on the lower right?
No, it's a great, right. Yeah, no, it's a great question. No, I actually don't know. The other thing I was going to add to you as well is that we only looked for OC43 specific responses. It could be so there's cross-reactivity against some of the other endemic coronaviruses as well, which we don't know about, and we didn't test for in the paper as well, partly actually because we didn't have enough samples to test for it. So it could be so as cross-reactivity against, for example, HQ1 or some of the other endemic ones, actually. But we don't know at this point if it's like that or not.
Thank you.
Is there any other questions? Oh, see one raised hand. Sean Murphy, I've unmuted you. Sean? I'm trying to unmute Sean.
It's fine.
Sounds like it. I think You can hear me now, right?
Yep.
That was a great talk. Thank you very much. I've been interested in resident memory T cells in malaria, and then of course I've been keeping an eye on things in SARS-CoV-2. So there's been a lot of talk about generating T cell vaccines for SARS-CoV-2 or pan-coronavirus vaccines along those lines. Given all the work you've done, I guess I'd just like to hear what you think about that as a strategy, given where we are in the pandemic. Do you think that that's a viable approach?
I think it could be a great approach together with actually the type of vaccines that we do have now. A combination. I actually thought about that. It would actually be a pretty cool like project grant almost to do to, that you basically give people or mice or even primates both maybe a shot in the arm and potentially nasal vaccines as well, just to see how that's going to boost both systemic immunity with, for example, antibodies, but also with TRMs. So I totally agree with you, and I personally I think that's the way forward for future vaccines, to administrate them, for example, through nasal routes, so that you can induce not only better resident memory T cells, but potentially also B cells in the future. So I think that will be great, and particularly to strike against the different variants of concern, of course. So I'm a fan of that idea. I have to be honest and say, yeah.
Great talk. Thank you.
On a related note, so I was curious what you think about the people who had this pre-existing immunity. Do you think that they could have been potentially exposed and just not acquired it as easily, like given the same number of exposures, or would they just get less sick?
Yeah. So at least in our case, of course, we are looking at people which were infected or they were not infected, so to say. This was before the pandemic, so they had these responses before the pandemic. But like I touched upon a little bit before, it was a little bit weird, and this was something that we started to see already at that time point, that some people that were exposed to the virus, it looks like they had higher levels of SARS-CoV-2 specific responses. At one point, back in the days where 1.5 years ago, I was pretty sure that meant that they were probably infected, but maybe they didn't seroconvert. Now I rather probably think that they just, again, they maybe wasn't infected, but maybe that boosted somehow, maybe their T cell response, maybe their cross-reactive T cell response as well. So it is hard to know, like it's a chicken and egg question, which is a little bit tricky to look at now. But I do firmly believe that if these responses are going to have a major role or play a major role, most likely they're going to have this sentinel type of response as we're showing here. If that's going to be enough with just some gamma to maybe, for example, to trigger an early, almost like innate like immune response with elevated interference in general, hard to say, but that's one of the theories or hypotheses that we have with some of the work that we're going forward with. Yeah.
Cool, thank you. Larry Corey has a question. Why aren't tissue resident memory T cells having a greater impact on replication long term?
In SARS-CoV-2 infection or in general?
No, I'm talking about HIV, not SARS-CoV-2.
Yeah. Okay. Yeah. Sorry. Yeah. Well, I guess it's hard to say. Of course, they do exist in most individuals, but at some point, I guess that they become exhausted as well if you tip that iceberg too early on. So even it's interesting because the TRMs also kind of become exhausted, and are less exhausted when of course in individuals which are controlling the infection. So they're there in all individuals, but the difference there between individuals controlling it versus not is really this fact that they're more functional in general, the TRMs in individuals controlling it. And it's not, it also has a little bit to do with the magnitudes there, as I showed, where it looks like levels are a little bit higher in the elite controllers, at least in the individuals that we looked for. But there are interesting features with some of that data that was generated. That's hard to say exactly what it's all about, but it seems to be that elite controls have TRMs which has much better capacity to rapidly, for example, translate mRNA into protein. It's not only cytotoxic abilities, which I think is a correlate to protection. It seems to be maybe other mechanisms as well. But again, to answer your question, it's about, yeah.
So
it's
you're
not-
sort of saying you think when exhaustion occurs, it's sort of a permanent state, and there's no way to unexhaust a population of T cells, and there's something unique about the retained functional activity, and it's not necessarily killing, it's either cytokine or proliferation that, or some other metabolic issues that define this. And if that's the case, what are you pursuing this, and what do you think mechanistically, how does that occur? How does that happen? And why would something be functionally exhausted forever, especially if they're on ART?
Right. So there are some work which is starting to come out, and I think particularly in the cancer field that it has been coming out that exhausted cells, they have this epigenetic imprint basically on them, which means that even if you try to block them with PD-1 inhibitors, they will divide, they will do to some degree, but it's not going to last forever. These cells, and even in ART treated individuals, there is something which is happening early, probably in HIV specific cells, that gives them this epigenetic imprint basically on these cells. That's what I believe, and makes them less capable to proliferate or be polyfunctional. And some of these things are the stuff that we are working on right now in gut sections, for example, trying to look in through the gastrointestinal lining, really trying to understand how that differs at different sites and between different epitope specificities. So we are pursuing it, but it's expensive technologies and hard to get the samples to pursue these questions in great detail. Yeah.
Helpful. Thanks.
Thanks.
Thanks. Next question's from Jazu. She says, great talk. Would you elaborate on vascular resident memory T cells? Their similarity or difference from those of specific tissues? And I'll unmute her in case she has a follow-up.
Yeah, great. I usually call them vascular resident cells, particularly I think for this talk, because I was talking about resident memory T cells in general. So there are some data in the mouse literature which goes in line with this as well. There's some work from Ulrich von Adrian's lab, Carmen Gerlach, which has been showing that these cells are expressing really high levels of the fractalkine receptor. So it's called C301. This receptor is actually not expressed on cells which are present within tissues. And then the question comes down to why, and it's actually true, it's not only for CD8s, it's for CD4s and for NK cells as well. So these cells which are harboring these really high levels of cytotoxic molecules in humans, they are present in the blood to a high degree, and they have a similar almost transcriptional signature to some degree, which is actually some things that we are also pursuing. But nevertheless, they're different from resident memory T cells in general, that can have other grand signs that they're expressing. And in some tissues, like the lung, for example, they might have the mRNA for peripheral integrin B expression, but they need a trigger of some type to upregulate it. And probably the reason for why they are not expressing cytotoxic molecules to a very high degree, at least peripheral integrin B together in tissues, is maybe because it's to not induce collateral damage basically in tissues. And the second question kind of relates to like, okay, but if you have these cells which are vascular confined or residency or what you want to say, well, what do they do there then? Why do you have cells which are only present within the blood? And basically there's two or maybe three different ideas about that. One, maybe they surveil endothelial cells, because many of these cells are going to be, for example, C and B specific cells. C and B can, for example, divide in endothelial cells. So maybe they are the vascular patrolling cells that are those cells that are, again, patrolling the vasculature to see if any virus is slipping out basically from the endothelial. Could also be so that they're penetrating through like fenestrated endothelium, like for example, liver and kidneys, so that they can kill target cells, but they can go back again. And the final thing could be that we don't really see, and people and research have not really seen in the mouse field as well, is that they kind of function almost like neutrophils in some ways, so that they almost have the blood as a reservoir, and when inflammation or something else takes place, they go to that tissue, and that's when they function. It could be a combination of all this, it could be one of them, but it seems to be that they are unique and different in some ways, at least from the cells which are surveilling the tissues, and definitely different from the resident memory cells in tissues in general. Yeah.
Thanks. So you think there are more kind of like blood circulating cells, but have features that can really quickly respond to infiltrate to the tissues?
Maybe, because you're expressing some specific, they are expressing certain chemical receptors. They don't have like these classical chemical receptors like CXCR3, CXCR6 to a high degree, but they have other ones as well. More of these so that they can home into endothelial cells and maybe also respond to stress from endothelial cells. So yeah, two different ideas. Either they surveil endothelial cells within the vasculature, or they respond to, for example, fractalkine when it's going to be produced by during inflammation and other cell by tissue damage, for example, and that's when they go out into tissues maybe. But honestly, we don't know at this point. The mouse field is more going in favor of that they're surveilling endothelial cells, but we don't know for sure.
Thank you. Yeah.
Welcome.
Thank you.
David Cole had one more question. Because if there's time, there was a recent scientific reports paper covering recovering CD8s from accessible nasal swabs, which looks doable. Have you looked at convalescent persons for nasal memory?
No, we haven't. I think it's a great question. One of the major reasons why we didn't do it was because it was going to be, we actually have some collaborators which is doing it, and they had a story in JCI on myeloid cells, and we tried it a couple of times to do stainings, for example, of SARS-CoV-2 specific cells. We had a hard time to recover enough antigen specific cells to do any meaningful analysis on it. So I have to be honest, say that we dropped that specific cohort, but we have had some ideas. Particularly what we're doing now is to follow up people that are going through tonsil surgeries. We have organ donors where we can look into all kind of organs in people which has become vaccinated versus non-vaccinated, unexposed as well, to really try to nail down and follow up on how these cells are functioning, potentially even from one tissue site to another versus blood.
Thank you so much. I don't see any other questions. I guess we can end the seminar early and let people grab coffee or something. But thank you so much, Marcus, for joining us.
Thank you. Pleasure's my side. Okay.
Take care, everybody. See you at the next CFAR seminar.
Yeah, thanks.